So I'm very glad to come here, and I'm going to talk about some of the recent statistical methods our group has developed for analyzing spatial transcriptomics data. So please feel free to interrupt me if you have any questions. Let's first go through some background. So the spatial transcriptomics is a very new technology. So if you have been in this field for a while, you know, the transcriptomic studies, it initiates from the bulk RNA sequencing studies. So bulk RNA sequencing study was early technology developed about maybe 10, 20 years ago. And so where with the bulk RNA sequencing, you basically can collect the tissue from an individual, and then you can measure the gene expression level in the tissue. Now, one main issue with the bulk RNA sequencing is that the tissue is really heterogeneous and contains of different cells from potentially very different cell types. It's essentially a mixture of different cells and cell types. So therefore, while bulk RNA sequencing is relatively cheap to do, you can collect multiple different samples. It does not have the cell type resolution or spatial resolution that can allow you to answer many different interesting questions. So in terms of analogous on that, you know, if you think about painting, the bulk RNA sequencing, it's essentially like you're mixing different colors together and put on a paint. Because of those limitations, the single-cell RNA sequencing studies have been recently developed. That was from 5 to 10 years ago. So single-cell RNA sequencing, now you are able to extract every single cell from this tissue and measure gene expression level for every single cell. So in terms of painting, it's like you're now able to sort out those different colors, and you can sort it out into different palettes. So that gives you a very high cell resolution. However, this single-cell RNA sequencing study does not give you spatial information. It does not tell you where each single cell is located on. So therefore, the spatial organization of single cells is actually quite critical for carrying out the tissue and complex tissue functions. And because of that, the recent technologies, multiple technologies are developed for collecting the spatial transcriptomics data, where the spatial transcriptomic data allows you to collect gene expression with the tissue, with the position spatial information. So in terms of painting, now you are able to put the right color into the right place in order to form a paint. And there are many different technologies have been developed. There are two general categories. One categories are sequencing-based spatial transcriptomics techniques, and the other categories are SN-FISH-based or single molecular fluorescent in situ hybridization-based technologies. So for the sequencing technologies, there are well-known spatial transcriptomics, which is precursor of 10x Genomics Visium technology, and 10x Visium, which is the most widely used commercial platform, and Genomics, that's another commercial platform, Statistical high-definition spatial transcriptomics, and more recently, seq-scope, stereo-seq, those very high resolution spatial transcriptomics. On the other hand, for the SN-FISH technology, we have MERFISH, which is now being commercialized. Also seq-FISH, seq-FISH Plus, COSMIX, and also the recent 10x Visium technology, the 10x from the 10x company, and that just came and become available from last October, November-ish. So let's take a look at one example from each technology just to see how the data are collected. So the most common platform, commercial platform, is the 10x Genomics Visium technology. So for this particular data, in order to collect this type of data, what you do is you can obtain the slides from the company, and on the slides you have 4 different square positions, and on each position you can overlay your tissue slides on in order to measure the gene expression level. So for example, if you are interested in measuring gene expression for the mouse brain, so you can cut half of this mouse brain and overlay on top of the square. And this square is very unique because the capture area contains a few thousand, 5,000 barcoded spots over there. And each barcoded spots contains a bunch of probes that allows you to measure the gene expression in a localized fashion. Specifically, you know, if you zoom in, you have a lot of spots. If you zoom in on each spot, you can see that this read contains 3 different components. One is a poly-T, which allows you to bind the poly-A that's released from the tissue in a very spatial localized fashion. The second part is a UMI. This is the unique molecular identifier, which allows you to remove the PCR bias during the process. And finally, it also contains a spatial barcode, which is very critical because once you sequence the barcode, it basically tells you where this probe comes from, in which particular location it comes from, and its X and Y coordinates. So therefore, once you collect the data, you essentially ended up with two measurement matrices. One is our standard N by P gene expression count matrix, where N is the number of spots you have, and the P is the number of genes you have. So this essentially measures the gene expression level on each of those unique barcoded spots in a very localized fashion. So this is a standard matrix we can also get from bulk and single cell RNA sequencing. But in addition to that, you also can obtain N by 2 location matrix, where it gives you the X and Y coordinates for each of those N different spots. So therefore, now you have the gene expression matrix and the spatial location matrix that allows you to combine these two to perform important analysis. So this is the 10x Visium technology. Another technology is the sequence hybridization-based or single molecular FISH-based. So one representative example is the seq-FISH Plus. It basically measures the gene expression in a sequential in situ hybridization process. And that way it allows you to measure typically hundreds of genes or sometimes even tens of thousands of genes. And one unique benefit of seq-FISH Plus or those single molecular first in situ based technology is that it gives you a very high spatial resolution. So you can actually detect gene expression at the cellular, at the single cell level. And at the end of the day, when you perform those SM-FISH-based technology, you are also going to end up with two measurement metrics. One is a gene expression matrix. It's an N by P count matrix, counting the number of molecular falling in each one of those N single cells. And you also have the N by 2 location matrix, where you can measure the X and Y coordinates for each of those single cells. So regardless of which technology you have, you are always going to get these two metrics. One is the N by P count matrix of gene expression. The other is N by 2 location matrix. And certainly the sequencing-based techniques and SM-FISH-based techniques, they all have their own benefits and also drawbacks. So for example, sequencing-based technology, it may tend to measure relatively lower number of spots, higher number of genes, relatively low spatial resolution, although newer sequencing-based one are going to give you very high resolution as well. And SM-FISH-based techniques give you a high number of spots, low number of genes, and relatively high spatial resolution, giving you the single cell resolutions. So those are the basic data features. And with the spatial transcriptomics, this relatively new technology, now you can start to answer a lot of different scientific questions. So here I'm going to talk about 3 different methods we developed to answer some of those important scientific questions. So the first, I'm going to focus on a method called Spark to perform SE analysis to obtain, to perform calibrated powerful SE analysis, where to identify those genes shows the spatial expression pattern. So the SE analysis, many people also call it to detect genes, detect SVG genes or spatially variable genes. So the question looks like this. So we have if you look at the figure on the right-hand side, so I'm showing here is the spatial transcriptomics data visualization collected from an early spatial transcriptomics technique. So this is on a mouse olfactory bulb. And the mouse olfactory bulb is symmetric. So you have 2 bulbs, one on the left-hand side, one on the right-hand side. And each dot represents a measurement spot. So you are measuring gene expression on each spot, and each spot allows you to measure tens of thousands of genes. And here, the color represents the gene expression level for one particular gene. So I'm only visualizing one gene. And over here, you can see that this particular gene, as the purple means high expression, blue means low expression. So for this particular gene, it's heavily expressed in the middle of each of these 2 olfactory bulbs, and relatively low expressed in the surrounding area of the olfactory bulb. So this gene shows this unique spatial expression pattern, and this differs from potential housekeeping gene where you might expect to show a sort of uniform distribution across the entire space. So our goal here is try to identify genes that show this type of spatial expression pattern, which then can give us a clue on how the tissues are really organized and how the transcriptomics really contribute to the functionality of the tissue. So when we first developed methods, there were two methods have already been developed. One is called TransSeq, the other called Spatial DE. Both methods have important limitations. So specifically, both methods, they try to model normalized data instead of the raw count data. But it's modeling normalized data, we all know, instead of raw count, we know that it's going to lose power. And the second main issue is that the p-value computation for this type of models is actually quite challenging. So for TransSeq, it's using permutation-based approach to compute p-value. So it's extremely slow. So it does not apply to even 10x Visium data. It can only handle 200, 300 spots. On the other hand, Spatial DE is using a fast approach to calculate p-value using an asymptotic normal approximation, but it's not accurate. As we'll show, it gives you inflated type 1 error control. And so therefore, motivated by these limitations, we decide to develop a method, SPARC, or Spatial Pattern Recognition by Kernels. So the main idea is this. So we're going to examine one gene at a time. We're going to detect each gene to see whether it shows spatial expression pattern or not. And for each gene, here are their notations. So the S sub I, it denotes the spatial coordinates for the I's location or I spots. So you have X coordinate and Y coordinate. And then on this measurement, measure the spot or measure location, we can measure gene expression level. And for this particular gene, we have Y sub I as a gene expression measurement count. So the Y sub I is the function of that location and it's a count measurement. And these two are the most important part. And certainly we also have a read depth, which we need to control for, which is denoted as N sub I, and X sub I, which has observed explanatory variables that sometimes we want to control for. But these two are less important. So the key part is the SI, the location, and the YI, the gene expression count. And because YI is the count, so we naturally model it through the over-dispersed Poisson distribution, where we assume yi follows the Poisson distribution with the red parameter as a product of n sub I times lambda sub I, where n sub I is the read depth normalization factor, while the lambda sub I is the important red parameter. And because lambda sub I is positive, so we model it at the log scale, and we model it as a combination of 4 different terms. The first 2 terms are standard terms. Specifically, the first term is just the intercept. The second term are the covariates you want to control for. But the next 2 terms are important terms that allows you to identify genes that show spatial expression pattern. Specifically, the epsilon I, it follows IID normal distribution. So the epsilon I is standard IID normal distribution, and we just use it to model over dispersion, the variance that's not explained by the Poisson model alone. On the other hand, the BI is critical. So the BI is try to model the spatial expression pattern. And to do that, we assume it follows the zero mean and the stationary Gaussian process, try to model the spatial pattern across the locations or the spots. So we have n different locations, so you can actually combine all the n b sub i's together into a b1, b2, b sub n by 1 vector. And for this entire n by 1 vector, we assume it follows a multivariate normal distribution with mean 0 and a covariance term that's characterized by a K matrix here. So the K matrix is a kernel function of the spatial location. And the K matrix here is try to model the spatial expression pattern of the B's. So specifically, if you consider 2 locations, I's location and J's location. If I and J's location, they are close to each other, then the K sub ij is going to be large. And so therefore, the B sub I and the B sub j is going to be more similar to each other. So this way, you try to introduce the gene expression similarity on the neighborhood locations. And so therefore, the B, the entire vector, captures this type of spatial expression pattern. And because the B captures the spatial expression pattern, to identify genes that shows the spatial expression pattern basically turns into a hypothesis testing framework that you only need to test whether the entire vector B is 0 or not. If the entire vector of B is 0, that means that you only have residual error, I, D normal distribution over dispersion, so there's no spatial pattern. But if the B sub I is, the B vector is non-zero, then it indicates that the gene expression is going to vary depending on their similarity or distance between the pairs of locations. So therefore, testing identifying genes shows a spatial expression pattern now is equivalent to testing whether the entire vector of B equals to 0. And testing the entire vector of B equal to 0 is also equivalent to testing the tau equal to 0 or not. Because when tau is 0, then the covariance is 0, so every B becomes 0. So therefore, now we can turn this scientific question into a statistical question of testing that various component tau sub k here. And certainly, an important part here is the K matrix, the kernel matrix, because the kernel matrix basically determines what type of expression pattern you are going to detect and how powerful you are going to detect it. And because for any biological data, you are going to see all kinds of spatial expression patterns. So therefore, instead of using just one K, we actually use 10 different Ks, try to capture a wider variety of spatial expression patterns. In particular, we have 5 Gaussian kernels and 5 cosine kernels. The 5 Gaussian kernels have different bandwidths to model how the gene expression similarity decays across distance, while the cosine kernels have five different periodicity parameters, modeling how they change, how the periodicity patterns vary across the entire tissue. So therefore, we have 10 different kernels. And for each kernel in turn, we basically perform the hypothesis test of whether there's a spatial expression pattern or not by testing the null hypothesis that tau k equal to 0. And because those are variance component models, we need to perform inference based on penalized quasi-likelihood, and we use the average information algorithm to form scalable inference. And to further speed up computation, instead of doing a likelihood ratio or score water test, we actually use a score test statistics for each kernel. And we need to compute a p-value based on a mixture of chi-square distribution in order to get the exact p-value. And so this way, for each kernel, now you get a p-value, but we have 10 different kernels. So we eventually need to combine all the 10 p-values through the Cauchy combination rule in order to arrive at a single p-value for each gene. So therefore, once you go through the entire process, you are going to get a 1 p-value for each gene, and that p-value is going to tell you whether the gene shows a significant spatial expression pattern or not. So to summarize this entire process, you can see that our goal is from the left side to the right hand side. Our goal is to detect genes show those spatial expression pattern. And to do so, we use the generalized linear spatial model, and we try to focus on testing the tau1, the tau parameter, the variance component parameter here. And to do that, we introduce 10 different kernels, and for each kernel, we can compute the exact p-value. So we get a P1, P2, up to P10 for each gene. And then we combine the 10 p-values together into a single p-value for that gene. So this is the entire procedure of Spark. So I'll skip all the simulations we have done, or a bunch of different real data we have done. We'll just focus on one example. So this is the human breast cancer data example. It's collected through the spatial transcriptomics sequencing technique. And we did a very simple filtering. So remove the genes that are expressed in less than 10% of the spots, and retain the spots with at least 10 total read counts. So we analyzed a total of about 5,000 genes measured on 250 spots. So here shows the results. On the left-hand side, it shows the type 1 error control. On the right-hand side, it shows the power. So for the type 1 error control, so what we did, it was just simply permute the order of the Ys. So therefore, it gives you a null distribution, so you can apply different methods over there, get that p-value. And if the p-values are calibrated, if they give you correct type 1 error control, then they should sit on the diagonal line. And so you can see that our method, which is the purple colored one, it does sit on the diagonal line, while some of the Trend-seq versions, it also sits on the diagonal line. But some of them are perhaps more conservative below this line. But for spatial DE, you can see that it's initially conservative, but later on, it goes above the diagonal line, indicating you're going to get a lot of false signals in the permuted null data. And with the permuted data, it also allows you to estimate the false discovery rate so that you can compare power for different methods versus based on the false discovery rate. So that's showing on the right-hand side. So you can see that our method is much more powerful compared with spatial DE and Trend-seq. So when we initially see these results, we were actually a bit worried because our group was mostly previously developed methods for GWAS, and we know that we're usually lucky if you get a method have a 10% or 5% power gain compared with others. And this time you see like 2 times or 3 times fold higher, more powers. So we decided to carefully examine all the genes that detected by our methods and versus other methods to make sure that we are actually detecting true biological signals. So the first thing we did was to look at the original study. And in the original study, they emphasized the 10 known cancer genes. And so we look at how many among those 10 cancer genes we can detect. So it turns out that SPARC can detect all the 10 cancer genes, while the other 2 methods can detect 7. So this shows the expression pattern. So on the left-hand side, it's actually a standard image, where the bottom, the dark area are the cancer regions, and the top white areas are non-cancer regions. And you can see that for the majority of those cancer genes, they do show a very high enrichment in the cancer regions, either entire cancer region or sub region of the cancer domain. And the second validation we did was to look at the overlap between different methods and simply ask whether the different genes detected by different methods, they do contain biological signal. So specifically, we examine the overlap between, for example, our method and the spatial DE. So when you do that, you can see that we detect 277 genes in total, and among them, 75 are overlapped with spatial DE. And we detect 202 unique genes, while spatial DE detects 30 unique genes. So therefore, we can group the genes into 3 groups. One are the overlapped group, and the other 2 are the genes uniquely detected by our method and uniquely detected by spatial DE. And then for those genes, we simply ask how many among them, how many among them they contain those known cancer genes. So we look at 2 cancer databases. One is the CancerMine, the other is Harmonizome. So in both cases, you can see that the overlapped set, you do have a higher percentage. For example, when you overlap with Harmonizome, about half of the 75 genes are well-known, previously known cancer genes. But for the genes detected by our method, it's still about 44%. Those are overlapped with the known cancer gene database, while for the genes detected by spatial DE, it's slightly lower, it's about 37%. So it indicates that the genes that are uniquely detected by us, they still contain a quite substantial proportion of those known signals. And so we can look at some examples that are only detected by our method, but not the other methods. So the first known example, very well-known example, is ERBB2. So this is a very well-known transcription factor for breast cancer. And our method can detect it, and it can show its special expression pattern that's enriched in the cancer region. We also detect HLA-B and CD44, both are immune cell markers. It indicates that immune cells are potentially infiltrated this cancer region that are potentially try to fight back against those tumor cells. And then we also have our EF1A, which is the elongation factor, and also MMP14, which is the extracellular matrix protein. The expression of both suggests that there's a potential extracellular matrix cleavage going on or migration going on, that the cancer is potentially in the later stage of development and becomes migratory to invade other tissues. So therefore, those examples, they all make sense, and they give you a very nice biological interpretation or insights into the cancer genesis. And finally, for the other genes, we also perform the gene set enrichment analysis. You can see the top GSEA pathways detected, they all make sense. They're all related to extracellular structural organization or collagen or response to cytokine or regulation of immune responses. So, so that's basically what we have for SPARC. So when we developed SPARC, it was about a few years, that was 2018. So that's 3 or 4 years ago. And after we developed SPARC, the field of spatial transcriptomics is really fast developing. So there's a lot of new data types coming out, and those new data types gives you a much higher resolution, spatial resolution, but it also gives you much larger data sets that create new challenges. Specifically, those new data types have increased sample size, And also, you tend to see a lot of low counts and prevalence of zeros in the data, and both creates computational and statistical challenges. So specifically, like the slide-seq, HDST, or more recently, stereo-seq or seq scope, they are of very high resolution. They can easily measure tens to hundreds of thousands of locations. And so the sample size becomes really large in terms of the number of locations you measure on the tissue. And because of many of those technologies are sequencing-based, and the sequencing-based technology are always limited by sequencing depths, and the increased data size resulted in a sparse form of the data. So in the N by P count, gene expression count metrics, you are going to see a lot of low counts and a lot of zeros. For some data, sometimes you can even see 99% of the entries are zero. And both creates important challenges. So in particular, in terms of increased sample size. So unfortunately, although Spark works well, unfortunately, Spark, along with all the other existing methods at that time, they all have a cubic computational complexity and also cubic memory requirement. And so this makes them very hard to scale to very large data sets that are collecting nowadays. So it can take days or months with dozens to thousands of GB memory requirement in order for them to analyze some of those recent technologies. In addition, the low counts and prevalent zeros are also creating important issues. Specifically, now if you have low counts, you tend to use a counter model to model them. Using counter model to model those sparse counts is very challenging in terms of computation. So it can lead to a failure of algorithms over a large percentage of genes. So even if you simulate it, and you can quickly try this. So even if you simulate data from, say, a negative binomial model, and as long as you keep the negative binomial rate parameter to be very low, so that is simulate a lot of zeros, and then you try to fit it with the negative binomial model, say a standard R package. To do that, you will see it will fail over 90% of genes. Simply, the algorithm is not that stable once you have a lot of zeros. But on the other hand, if you try to model the count data with normalized model, with normal distribution, it also is problematic because approximate modeling of those sparse count usually results in both power loss and failure of type 1 error control at small p-values. So at that time, we actually tried to build a Gaussian version of Spark to model the normalized data, but it's not working well, just like the spatial DE I'll show you later. Both will have power issues and have type 1 error control issues. So motivated by this, we decided to develop another version of Spark, extended version of Spark, which we call Spark eXpedited. It's actually using a completely different framework to perform hypothesis test, but the Spark X is extremely scalable. So Spark X is based on a general class of covariance tests and for non-parametric spatial expression analysis. So the main idea is this. So we're still going to examine one gene at a time. And for each gene, we're going to create two kernel metrics. One is the expression kernel matrix E. The other is the distance kernel matrix sigma. And so if you think about two locations, I's location and J's location. So if I and J's location, they're close to each other, then the sigma ij, the corresponding element in the distance kernel, is going to be large. And if I's and J's location, they show similar gene expression patterns, then in that expression kernel matrix E, the eij is also going to be large. Now, if you want to identify genes that show spatial expression pattern, you basically want to see, to show that the sigma, the ij and eij, they are going to be correlated with each other. So if you have a high sigma ij, you want to see a high eij. So that's the main intuition. And so to do that, you can compute this trace term, E times sigma, and this trace term under the null, it will follow a mixture of square distribution. So that allows you to get a p-value to test whether the gene shows spatial expression pattern. And certainly it's, we need to do a lot of engineering in the process in order to make it actually work well and work fast. Specifically, we need to perform a range of nonlinear transformations on the x and y coordinates in order to capture a wide variety of possible spatial expression patterns. And then we need to combine all the p-values through all those different nonlinear transformations, again, through the Cauchy combination rule to get a single p-value. And we also need to deploy several important algebra tools and manipulations to ensure the method is really highly scalable. So at the end of the day, the algorithm and the method is quite impressive. So it has a computational complexity and the memory requirement. Both are linear with respect to the number of spatial locations. So it's going to be n squared times faster than any of the previous methods. And n is typically, in the range of easily thousands. So you can expect it to be 1 million times faster to compare with the existing approaches. So we can take a look of the computational gain. So over here we see two plots. One is showing the time versus sample size on the left-hand side, and on the right-hand side we have memory requirement versus the sample size. Well, the sample size means the number of measured locations on the tissue. So you can see the spark, which is a blue dot. It's very powerful, but it's not that scalable. So you can see that it's very slow. So you can handle thousands of locations, but once it's exceeding, say, 10,000, 20,000, it's getting very slow. And the Gaussian version of Spark, which is the green line, works pretty well. And the Spatial D also works fine. But they both show the cubic increase. So therefore, they can scale to about 20,000-ish. But once beyond that, it's going to become very, very slow. And however, for SparkX, you can see that it's a linear increase pattern. So on this plot, you don't even see much difference when you analyze 3,000 versus 50,000. It just takes less than a minute to analyze as it's a very large sample. And in terms of memory, you see the same pattern. So SparkX is extremely computationally efficient. So with this, so we applied SparkX through comprehensive simulations and also multiple data sets. So we'll talk about a couple of them. So the first one is the mouse cerebellum data. So it contains 11,000 genes measured on 25,000 spatial locations. And so the cerebellum looks like on the top left corner. So you have an illustration of how the cerebellum looks like. It's very well organized structure. It has about 10 different lobes. They show these lobular structures. And so when we analyze the data, we first measure the time and the memory usage. You can see that as in simulations, SparkX basically takes a very small amount of time and uses only a little bit memory compared with the other two methods, the Gaussian version of Spark or the Spatial D. And in terms of type 1 error control, you can see that SparkX sits on the diagonal line. The Gaussian version of Spark gets inflated because it's using model the count data with normalized with Gaussian distribution, while Spatial D also even more inflation. And certainly in this case, if you apply Spark, it will still sit on the diagonal. It's just not that scalable. And in terms of power, you can see that SparkX is more powerful than the Gaussian version of Spark and the Spatial D as well. And certainly if you apply Spark over here, it will be more powerful than SparkX. But it's just scalability is the main concern with Spark. And we can do all those standard overlap analysis as well. So here shows some examples. You can clearly see that they see the spatial expression pattern and they seem to mark a particular cell types. So when we see that, we decide to, it gives us some thoughts, what are those spatial expression genes and why genes show spatial expression pattern. So at that time, one hypothesis we had was that the genes that show spatial expression pattern, potentially because many of those genes that are cell type marker genes. And we all know that cell types, they tend to show a different distribution on the tissue. And so therefore, if genes are cell type marker genes, then they are going to label those cell types. So they are going to show spatial expression pattern simply because the cell types shows a spatial distribution. So we decide to test whether that's true or not. So it turns out it's to be half true. To do that, we look at another mouse cerebellum data sets from Statistical version 2. So we have 20,000 genes measured on 11,000 spatial locations. And we did 2 SE analysis. One is our standard SE analysis, try to detect the SE genes. The other is we introduce the cell type compositions as covariates to control for the cell types. Intuitively, after you control for the cell types in the model, then anything you get are essentially the genes that show SE expressions that are independent of the cell types. So therefore, we did this 2 analysis. So in the first analysis, the regular analysis, we detect about 520 genes. So that's a big circle here. And then after for controlling for cell types, we still can detect about half of them. You can see still about half of them still remain significant, meaning that half of the genes that show spatial expression pattern likely not because of the cell types that show spatial expression pattern. So we decided to take a look at some of those examples. So here shows two examples. So the top, it shows, so the left column, it shows the cell type distribution on the cerebellum tissue. Specifically on top, the blue labels the granular cells, and while bottom, the blue labels the Purkinje cells. There's two different cell types in the cerebellum. And on the right-hand column, it shows two genes that shows these are cell type marker genes. On top, the CADM3, that's a granular cell marker. The bottom, the KELP1, that's basically the Purkinje cell marker gene. And you can see that they both show spatial expression patterns. So in the parentheses, you have two different p-values. So the second p-value is essentially the original analysis. You can see that they show spatial expression pattern, so their p-value are significant. One is 10 to the -7, the other is 10 to the -4. However, once you control for the cell types, you can see that their first p-value is no longer significant. That's because their spatial expression pattern matches exactly of the cell distribution of the cell type. So therefore, once you control for cell type, it's no longer significant. However, these middle two genes, are still significant even after you control for the cell types. So specifically the PTPRT, you can see that tends to express in only the frontier part of the granular cell population, only the left half of the cerebellum. While the LDOC basically is expressing the anterior, so the posterior part of the Purkinje cells, so the right-hand side of the Purkinje cell population. So therefore, for those genes, even after you control for those corresponding cell types, they are still significant, or sometimes even more significant. So this suggests that it's really important to detect the SC genes that are show spatial expression pattern after the just for cell type, because it gives you idea on the spatial compartmentalization of the cerebellum. So indicating that anterior and posterior part of cerebellum, they are potentially functionally different from each other. And finally, we applied to the SPARC-X to a very large data collected by HDST, which contains 20,000 genes measured on 177,000 spatial locations. On this data, none of the other methods would work, while SPARC-X only uses 0.4 GB memory and takes 3 minutes. And it's the only method that's applicable here. And all the results here, they all still make sense, and gene set enrichment gives you the top pathways are related to a synaptic connection or neuron synapse. Okay. So this SPARC and SPARC-X, they basically try to target to identify genes that show spatial expression pattern. And then now the next part we're going to talk about method card is for a different type of analysis. It's for cell type deconvolution. So specifically, the motivation is that many of those spatial transcriptomics are of relatively limited spatial resolution. So for example, the 10x Visium technology, that's the most widely used commercial platform. It measures gene expression on thousands of spots, but each spot is not at a single cell level, but instead each spot actually measures 2 or 3 different cells, or sometimes even more different cells. So therefore, it's measured in the average gene expression of those few different cells in the localized fashion. So once you have the data, if you are able to perform deconvolution, identify on that particular location, what are the cell types are over there and what's their percentage, and that will give biologists much more information on how the tissues are organized. So ideally, on each measured location, you want to perform deconvolution to tell, maybe there are 5 different cell types in this particular measured location. Maybe that 25% of them belongs to a cell type A, 50% belongs to a cell type B, and so on and so forth. So therefore, we want to develop a method to perform cell type deconvolution. And to do that, we can integrate this existing single-cell RNA sequencing studies into spatial transcriptomics in order to perform such type of cell type deconvolution. That's because the single-cell RNA sequencing data, they are abundantly available. You can easily find tissue that corresponding to the spatial transcriptomics. And in the single-cell data, the unique part is that you have the single cells. So you can get the cell type marker genes, you can get the cell type marker gene expression pattern on each cell types. And the cell type specific expression pattern basically gives you information to perform deconvolution on spatial transcriptomics. So when we first started the work, there was really only one method available for bulk RNA sequencing deconvolution. So that's the music. During the development of our method, the field is moving really fast. You can see that in the process, there are 5 different methods have already developed during the revision of our paper. However, those existing methods, they all have important limitations. Mainly, the first one is that they all ignore the spatial correlation among cell type composition in neighboring locations. So specifically, if ice and GS location are close to each other, you might expect the ice location and the cell type composition on these 2 locations are going to be similar to each other. And this similarity can actually allows you to borrow information across neighboring locations or even across the entire tissue to do a better job on performing the cell type deconvolution. So therefore, it would be actually nice to incorporate this type of spatial correlation structure among cell type compositions. The second important limitation of those methods is that they all need to rely on a single cell reference panel to perform cell type deconvolution. And it turns out that when we develop our method can either use a single cell as a reference, but it also can perform cell type deconvolution in a reference-free fashion so that you no longer really need to provide any single cell reference data. And finally, all those methods, they focus on deconvoluting measured locations, and they can be of limited spatial resolution. While our method turns out that because of its modeling of the spatial correlation, you can use it to impute cell type composition or impute gene expression on unmeasured locations. So, the method we developed is called CARD, or Conditional Autoregressive Based Deconvolution. So the main idea is based on this non-negative matrix factorization model. So here, X is the gene expression data, which is, I think it's P by N matrix gene expression. And the B here is the loading matrix or the single cell reference data. So essentially contain the cell type specific marker gene expression. And so if you have single cell reference, you can compute B. If you don't have a single cell reference in our model, you can infer B directly. And then the factor part, the V part, is the cell type composition. So the V part is our focus. So it's try to infer this N by K matrix where you have K cell types and you measure the cell type composition on each of the N locations there. So this is our basic model. So it's a non-negative matrix factorization model because it's non-negative on the V. So the V are constrained to be between 0 to 1. Now, for the V matrix, we specified a conditional autoregressive modeling assumption on the columns of V in order to model the spatial correlation across neighboring locations. So specifically, we are trying to say is that now the cell type composition on ice location is really depends on this neighborhood locations as well. So you can borrow the neighborhood information to better estimate it. So we use a count modeling assumption on the V in order to encourage that. Modeling such correlation is very important. Not only it can borrow cell type composition information across the neighboring locations, across the entire tissue so that it can get a better performance for deconvolution, but also it can help you impute cell type compositions as well as gene expression levels on unmeasured tissue locations. So therefore, it allows you to construct a very refined spatial map. And for this model, we developed a constraint optimization algorithm in the maximum likelihood framework to estimate that V matrix. So just to summarize the procedure of CARD. So we have our spatial transcriptomics data listed on the right-hand side. Our goal is to perform deconvolution on each of those measured location or measured spot. And we can use, if we have a single cell RNA sequencing data, you can use that as a reference. If you don't have that, it's still okay. We can do a reference-free deconvolution. And the model is based on the conditional autoregressive model-based approach to perform deconvolution, and we can deconvolute each measure location essentially into a cell type composition matrix. So on each measure location, we usually use a pie chart to represent the deconvolution results, where different colors represent different cell types, and the size of those pie charts represents the proportion of cells. And in addition to perform cell type deconvolution, you can also construct a refined spatial map in terms of cell type composition or in terms of gene expression through this imputation strategy. So we can look at one data here. So this is human cancer data. So the spatial transcriptomics measures 25,000 genes on 400 locations, and we did two analyses. One is reference-based, that's reference-free. So for reference-based, we use a single cell RNA sequencing data by InDrop, which contains 19,000 genes and 1,900 cells there. So this shows the deconvolution results. So if we look at card. So you can see that each dot represents a pie chart, and the size of the charts represents a different composition of the different cells. And you can see that the results from card and some of those methods, like for example, cell to location, they actually capture the histology annotation relatively well. However, unique compared with cell to location is that the card can actually detect two subregions of the cancer. So specifically, the cancer region is the top right corner. And in this region, you can see that it can detect two sort of subregions. In one subregion, the bottom region, it's mostly dominated by the pink cells, while in the top right corner, it's dominated by both the pink and the green cells. So the pink cell is a cancer clone B cell, while the green cell is a cancer clone A cell. These are two different cancer cell types that represent different stages of the cancer. So therefore, it indicates that this cancer region is heterogeneous and contains a progression from initial stage to a later stage cancer. And you can validate the cell types and distribution by looking at their distributions, different cell types on the tissue. And you can also quantify the accuracy by perform a correlation with single cell RNA sequencing data. So specifically in a single cell RNA sequencing data, you know the average cell type composition. And so once you perform deconvolution, you can also calculate the average cell type deconvolution composition from spatial transcriptomics, and then ask which one has the highest correlation with the single cell data. And so you can see that CART gives you a highest correlation. It indicates that at least on average, it gives you better results compared with other methods. And with CART, you can construct a more high resolution. So specifically, showing here, we see the resolution refinement for the cell type composition. So the top row shows the original resolution. In the bottom, we show the high resolution. So you can refine it. And we can do that because of the CAR modeling assumption, the conditional autoregressive assumption allows you to perform imputation on unmeasured locations. And then not only for cell type composition, you can also do this imputation and generate a fine map for the gene expression for each of those individual marker genes as well. And with CART, you can also perform reference-free deconvolution. And in the reference-free, certainly you use less information. So you tend to generate less accurate results compared with the reference-based approach, especially when the reference fits your spatial transcriptomics data well. So in this particular case, you can see that generally it works pretty well. It captures the general structure of the tissue. But in this particular cancer domain, you no longer see the subdomains anymore. And you can also examine computational efficiency. So the CART is very fast compared with other methods, and also uses relatively low memory. And so it's actually quite comparable with the bulk method music there. And so after we published our paper, there's a recent study from a different group. They did a comprehensive benchmarking study on different cellular deconvolution methods for spatial transcriptomics. And so this paper just came out last month. And we were really happy to see that this in this benchmarking paper, they found that CAD performance is the best among a range of different methods. Nowadays, you can see there's almost like 20 different deconvolution methods available. And in terms of the overall performance, CAD is the best one among the other methods from this independent separate study. So finally, I'll just summarize what we have talked about. So first, we talked about the SPARC and SPARC-X, which are effective methods for detecting genes that display spatial expression pattern. Then we also talked about CAD, which enables the spatially informed cell type deconvolution for spatial transcriptomics, and it can use either using a single cell reference for deconvolution or without a single cell reference to perform reference-free deconvolution. And all the three methods are available on our lab website, and their details are described in the three papers. And finally, we'd like to say that all the work really done by three trainees in our group, and one postdoc and two graduate students, Sichuan, Jiaqiang, and Yin, and they are the ones really leading the entire effort of the projects. And our all the projects are supported by NIH grants. So that's all I have today. I'm happy to answer any questions you may have. Thanks.
Thanks, Xiang, a very interesting talk. So now we are open for questions. So you can either just unmute yourself to ask the question, or you can put your question in the chat box if you like.